Good morning, children. Now let's start with the next topic, mitosis. Mitosis is a type of cell division which takes place in the somatic cells in the body cells, except in the reproductive organs. Children, we learn later there are two types of cell division, mitosis and meiosis. Mitosis is the type of cell division which takes place in the body cells or the somatic cells, and meiosis is the type of cell division that takes place in the reproductive organs at the time of gamete formation. Now today we will be learning in detail the process of mitosis. So mitosis is the type of cell division that takes place in the body cells or the somatic cells. As a result of mitosis, one cell gets divided into two, or two daughter cells are formed. The number of chromosomes in the parent cell and the daughter cells are the same. So it is called as equational division. Here we can see a picture which shows mitosis. As a result of mitosis, the parent cell gets divided into two daughter cells. The number of chromosomes in the parent cell and the daughter cells are the same, so it is called as equational division. The process of mitosis is divided into four stages: prophase, metaphase, anaphase and telophase. Children, by heart the terms prophase, metaphase, anaphase and telophase. One more time, children, repeat with me: prophase, metaphase, anaphase and telophase. Now, before we start with prophase, just recollect fast what all things we have learned about interphase, the structure of nucleus during interphase. Here we can see the cell during interphase. Interphase means it is the non-dividing phase. That means the cell is not undergoing division. During interphase, the structure of nucleus is very clear. It is covered by a nuclear membrane, a double-layered nuclear membrane which is having the nuclear pores in between. Inside the nucleus, you can see the spherical structures which are called as nucleolus, and the network of threads which are called as chromatin. The chromatin is present as a loose network of fibers which we commonly call it as chromatin reticulum. After interphase is the dividing phase or mitosis. The first stage of mitosis is called as prophase. During prophase, the first thing we can see, children, these chromatin fibers, the loose network of fibers, each chromatin fiber, it undergo condensation. It undergo coiling and condensation. As a result, each chromosome becomes visible. The chromatin undergo coiling and condensation and it becomes chromosome. You can see the chromosomes over here. Each chromosome is having two chromatids. Two chromatids are held together by means of centromere. Then we can see the nuclear membrane. Slowly it starts to disappear. Again we can see, children, during the S phase of interphase, we have learned the centriole duplicates. The centriole, which is already duplicated during the interphase, now it starts to move towards the opposite poles. Here we can see, children, these are the centrioles. These centrioles, it starts to migrate, it starts to move towards the opposite poles. Yes, children, one more time, let's go through in detail the prophase. During prophase, the chromosomal material, it condenses and forms chromosome. Each chromosome consists of two chromatids which are held together by centromere. The centriole, it moves to the opposite poles. Here you can see this is the centriole. It moves to the opposite poles. And from these centriole, from the centrosome, the spindle fibers starts to emerge from the centrosome. The nuclear membrane slowly it starts to break down. The cell organelles like Golgi complex, endoplasmic reticulum and nucleolus disappears. Here children, let's by heart fast what are the events taking place during prophase. One more time. The most important point, the first thing, the chromosomal material undergo condensation and each chromosome becomes clearly visible. Each chromosome consists of two chromatids which are held together by centromere. The centriole, which is already duplicated during the interphase, it moves to the opposite pole. From this centriole, the spindle fibers emerge. The nuclear membrane slowly it starts to disappear or it starts to break down. The cell organelles, the Golgi complex, endoplasmic reticulum and the nucleolus disappears. The second stage after prophase, the next stage is called as metaphase. During metaphase, that is the continuation of prophase, the condensation of chromosome is completed. That means each chromosome can be clearly visible. Each chromosome is made up of two chromatids which are held together by centromere. The spindle fibers attach to the kinetochore of chromosomes. Children, you have learned each chromosome consists of two chromatids which are held together by centromere. On either side of the centromere, there are disc-like structures which are called as kinetochore. So, on the kinetochore, the spindle fibers get attached. Now, these chromosome, it moves to the spindle equator, towards the equator, and they align along the metaphase plate through the spindle fibers to both the poles. I am repeating, children. The most important event during metaphase, the chromosomes. Each chromosome we have learned consists of two chromatids which are held together by centromere. On either side of the centromere, there are disc-like structures which are called as kinetochore. The spindle fibers get attached to the kinetochore. Now, during metaphase, the chromosomes move to the equator and they lie in the equator of the cell. This plane of alignment is called as metaphase plate. The next stage is anaphase. During anaphase, the centromere split and the chromatids separate. Here we have learned, children, this picture we have learned. This is the chromosome on either side of it consists of two chromatids which are held together by centromere. On either side of the centromere, we have learned there are kinetochore. Now, during anaphase, the centromere here, the centromere region, it split, and as a result, the chromatids gets separated. Now, the chromatid, it moves to the opposite poles. So, the most important point here, children, during anaphase, the centromere split, and the centromere split, the chromatids get separated, and the chromatids, it moves to the opposite poles. The last stage is telophase. During telophase, the chromosomes, they already started moving towards the poles. They reach the opposite poles and the reverse of prophase is actually taking place. What do you mean by the reverse of prophase? During prophase, we have learned the chromatin undergo condensation and forms chromosomes. During telophase, the chromosomes decondenses and it forms the chromatin reticulum again, the chromatin loosely arranged network of chromatin fibers. The nuclear membrane, it starts to reappear. Nucleolus, Golgi complex and endoplasmic reticulum reappears. Here you can see, children, the nuclear membrane, it reforms. The nucleolus, it reappears and the chromosome starts to decondense. Yes, children, here the picture, it shows the different stage of mitosis. Here the first one, it shows the interphase. During interphase, you can see the nucleus can be clearly seen. It is having a nuclear membrane. You can see the chromatin fibers and the nucleolus. The interphase is followed by mitosis, which is divided into prophase, metaphase, anaphase and telophase. During prophase, the chromatin material undergo condensation. Each chromosome becomes clearly visible. Each chromosome consists of two chromatids which are held together by centromere. The centrosome, it moves to the opposite poles. During metaphase, the chromosomes lie in the equatorial plane. The plane of arrangement is called as metaphase plate. Now the spindle fibers, it get attached to the kinetochore. During anaphase, the centromere break, the chromatids get separated and it moves to the opposite pole. And the next phase, the telophase, the chromosomes, they reach the opposite poles. The chromosomes decondenses, nuclear membrane, nucleolus, Golgi body, endoplasmic reticulum, everything reappears. Children, you should learn these diagrams